days about deep learning being applied in healthcare already. I want to add two more examples to that, coming from two companies that I work very closely with. So my first example is HeartFlow. So HeartFlow is looking at coronary artery disease, which is a pretty difficult or challenging disease and is affecting millions of people. It's actually one of the biggest killers in the world. So coronary artery disease happens when there's plaque that gets attached to the walls in your coronary arteries. And if this plaque becomes more and more, your vessels actually get blocked and the blood cannot flow anymore properly, and that might lead to stroke or heart attacks. The difficulty is in diagnosing coronary artery disease and also to decide which treatment option to apply. There are simple treatment options like changing your lifestyle, giving certain medication, or more severe treatments like intervention, putting a stent, and undergoing surgery. So how do we know which treatment option to apply? The problem is that the patients will show to the doctor with very similar symptoms of chest pain. So far, what is being done is to use invasive methods. So there is angiography, where patients have to go to the cath lab and they get a contrast agent and their blood vessels are imaged over time, and the doctor might identify whether there's obstructive coronary artery disease. Afterwards, they need to use a catheter with a pressure sensor to measure what's called the flow fractional reserve before and after the plaque, and that ratio tells you how bad the blockage is and whether a patient needs to undergo treatment. The interesting thing to note here is that most patients who go in the cath lab actually don't have obstructive CAD, so they actually could go with a simple treatment. However, this diagnostic task is very invasive and comes with risk and a lot of costs. So HeartFlow is looking at how to do this non-invasively by, for instance, taking a conventional CT scan, which we consider to be a non-invasive imaging technique to show anatomy. But how do we get functional measurements from that? That's where HeartFlow analysis comes in. They are able to what's called the FFR-CT, so they try to approximate the FFR value that you would get invasively by just doing this via a simulation from a CT scan. So this is how the HeartFlow analysis looks like. A doctor who has a patient coming in with chest pain would send in their CT scan. HeartFlow takes that conventional CT scan, extracts an anatomical model, gets some physiological measures that they need to run a simulation on the blood vessels, and then use computational fluid dynamics to simulate blood flow, which then can help to identify coronary artery disease and also measure how severe that is. So there were several trials being run for this already. The Pacific trial showed that compared to previous non-invasive techniques, the HeartFlow FFR-CT outperforms all other non-invasive imaging techniques. In the platform trial, which was a prospective multi-center clinical trial, it was shown that using the HeartFlow analysis on patients before they are sent to the cath lab can actually reduce the number of people who have to undergo invasive testing by 83%. That's massive. So there's a lot of cost saving involved, but also a lot of risk reduced for those patients. And for the patients who then didn't undergo the invasive measurement, there were no adverse events found. And of course, this can not only reduce the cost dramatically, but also help the patients by not having to undergo this invasive measurements. So the interesting thing about HeartFlow is they do use deep learning technology within their HeartFlow analysis. And it is already part of the clinical practice around the world. There are more than 20,000 patients who have undergone the HeartFlow analysis. There is reimbursement by the major insurance in the US. And interestingly for us here in the UK is that the NHS is helping a great way by enabling the pathway to use FFR-CT for our patients here in the UK. So first, in end of 2016, the NHS, the NICE guidelines suggest that coronary CT should now be used for patients with chest pain. So if you come into the hospital, you report about chest pain, it's very likely that you will get a CT scan. Now, a little bit after that, and after the results coming in about the effectiveness of the HeartFlow analysis, the NICE guidelines were updated in saying that for those patients where there is some uncertainty, the HeartFlow analysis is now the suggested analysis to identify which patient needs to undergo further treatment. And just recently this year, HeartFlow is now part of the NHS Innovation Technology Payment Program, which means that actually the HeartFlow analysis is rolled out at larger scale across the UK. We are very excited about this at Imperial College because also this year we just recently announced a very close research collaboration with HeartFlow, which means that the technology that we develop in academia can now be actually tested in the real world. So we are very excited about this, in particular about the deep learning aspects of this, which is our research focus at Imperial. The nice thing about the HeartFlow pipeline, if you look at it, is that there is this loop here where a deep learning technology can be used, for instance, to extract the anatomical model of the heart, and then this goes to an analyst. The analyst might correct those automated measurements and segmentations from the CT scan, but they also will feed back to the automated analysis where have corrections been done. So you have this very nice loop of the human-machine interaction, and you can imagine by acquiring more and more data, with a human in the loop, an expert in the loop, making sure that the automation is getting better and better. This is a beautiful scenario for applying any machine learning technique, but in particular deep learning. So this is a short video, a primer of something that HeartFlow is currently working on. Imagine if you have this 3D model of the heart, and you can make these measurements of fractional flow reserve, you can actually also simulate what would happen if you, for instance, do an intervention and might open up a vessel. What would happen to the patient's function of the heart? The other example I want to show you is Kieron. So Kieron Medical Technologies is a London-based startup that I work with, and they are experts in deep learning and develop deep learning-based radiology solutions. Their focus at the moment is breast cancer detection and mammograms. And the interesting thing here to note is they just got their first results from a clinical trial, and they're one of the first companies who actually got a CE mark for a deep learning-based radiology solution. And I will talk a little bit about Kieron in the following as well. So our journey in deep learning on the academic side started with something that we call Diabetic, developed by one of my PhD students, Kostas Kamnitsas. A couple of years ago, we started to look at how can we best analyze brain scans and how can we detect lesions automatically and how can we quantify lesions in brain scans. So we specifically designed this neural network, which is called Diabetic, which is designed in a way that it can make best use of the features that you find in a brain MRI to then predict at every point in this 3D scan whether that point is suspicious, whether it's part of the lesion or the tumor that you want to analyze. We got very good results with this over the last years. It got better and better. So last year, we won one of the international challenges, computational challenges on brain tumor segmentation. And I'm just showing you some visual results. So this is a brain MRI where you clearly can see the tumor, but it's difficult for a radiologist to measure that tumor, right? So it's especially to measure what the different parts of the tumor are in size and volume, how much they have already infiltrated the brain. So I'm now going through a few slides. So if I flick forth and back, what you do see is the automated results and the manual segmentation that a human expert would do if they would have sufficient time to actually do this tedious and time-consuming task. So here are some more results. And what you hopefully see is that there's not that much difference between the automated results and the manual results. You might even argue that sometimes the automation looks slightly better, a bit smoother. We used similar techniques like Diabetic in other applications. For instance, we used it for cardiac segmentation, 4D cardiac segmentation to get a 4D model of the heart. We used it for whole body organ segmentation. Now, these are all typical tasks where you can make use of automated segmentation techniques to do further analysis, like correlating anatomical phenotypes with maybe non-imaging information. Now, the problem in all these is that currently what we can assess is whether we achieve human-level performance. Right? So what we do is we compare how well does the automation do compared to what an expert would do on the image, right? If they could contour out the organs. So we did this for the cardiac case, and this was just published, so it just came out in JCMR, where we now can claim that our method is as good as the human. How do we do this? We actually look at automation versus human, and at the same time we look how do different humans do. So we compare different experts with each other. Now, our argument is if we get into the range of variability of making measurements, which is in the same variability as the humans are among each other, then we're done. Then we have achieved human-level performance. Right? So you might argue with that, but that is probably the best you can, how you can assess such an intermediate task like segmentation, whether you have achieved human-level performance, and whether you might actually, in practice, replace a manual expert that draws contours with an automated machine. Now, the question is, how do we go beyond? So how do we measure, or how do we assess whether we are maybe at superhuman level? Now, the difficulty is that we always go back to what the manual expert did, right? So we always compare to that. So what if we actually compare them, not like measuring how well we match the human, but what if we have a board of human experts looking at the results, not telling them which one is which, and making a call which they believe is better? So that's exactly what we did, human expert versus the machine. So we are very fortunate to collaborate with Ross Mitchell from the Mayo Clinic in the US and his colleagues on a study where we do exactly this. So we use our Diabetic network, or neural network, and trained it on 600 patients that they carefully annotated over a couple of years, where they basically did the manual drawings. We trained that neural network, and we tested it on 100 patients that we have never exposed to the algorithms before. And the interesting thing here to note is that this is really real-world clinical multi-site data as you would find it in any hospital. So it's sometimes messy, sometimes it needs some pre-processing, sometimes you deal with artifacts and so on. So this is really reflecting the real world. What we wanted to know is how good is the machine versus the human expert here. So what Ross Mitchell and his colleagues set up is a system where the experts could look at the two results side by side, and they had to make a call. And they did this quite extensively. So they invited 20 Mayo Clinic neuroradiologists. They looked at them at the manual and auto segmentation side by side. They were completely blinded. They didn't know which one is which. And they had to, each expert had to evaluate 20 out of the 100 cases. And they had to give them quality score from 0 to 10. Okay? Now, here are the results. Unfortunately, I can't share them yet. I'm sorry about this. We will release them very, very soon. But at this stage, let's assume that we were very happy with the results. And thinking about that, if it's true that our machine is better than the human, what does it actually imply? The machine has been trained on manual input, on human expert input, right? What it implies is if the machine is better, that it could actually outperform its human teacher. So the machine, by seeing enough, even like error-prone or subjective manual drawings, it could actually learn better to do that task. That would be the implication, and that would be big. So that would mean that we actually have achieved superhuman performance by actually using a human teacher. That would be nice. But then we can immediately ask the question, as an academic, I have to ask the next question, how do we go beyond that? How can we get even better than this? The problem, of course, is exactly what I just said. We are still training on manually provided segmentations. So they will always have errors. So maybe there's a limitation. Maybe there is an upper bar where that is the level of performance we can achieve. So last time I finished my talk at the Deep Learning Summit by asking the question, how can we go beyond, could we maybe synthesize ground truth? Would that be the solution? Could we synthesize ground truth data where we know the answer is perfect? There's no human error involved. If we train on something like this, could we get even better? So we have done a little bit of work on this, and this is work with the deep learning experts at Kiron, where we looked at, can we synthesize medical data? Can we synthesize high-resolution mammograms? So this project was led by Dimitrios Kokinov. I'm showing you some results here that I'm personally very excited about. One of those two images is fake. It has been generated by a computer. The other one is a real clinical mammogram. So you can tell me which one it is. It's very difficult, even for an expert, to say which of those two images is not true. Here are some more examples. So what we try to do here is to match some of the real examples that we have, roughly like visually matching to some of the computer-generated examples at the bottom. And you get an idea that the computer is quite good in generating those images. Now the big question is, maybe I'm just showing you some real images that have been somehow memorized by the network, and it just copies them and presents them as being new. But how can you tell if you have a large database of a few million of images, whether any of those is actually really in your database or whether it's something new? What you can do in this, and we are using deep learning technology here, which is based on generative adversarial networks, you can look at the space which you have mapped. So it's some new space where you have learned how these images are generated. We call it the latent space. You can sample in that latent space, and you can make smooth transitions. So I'm showing you now a video where you can see these transitions, and all of those images that you see on the left and the right are basically computer-generated medical images. What's interesting to note here is the huge variability that you find, the different sizes, the different textures that you find, and they're very much reflecting what you find in a large population of breast mammograms. Also interesting to note for the space of mammograms, you might have seen similar results where companies produce fake celebrity images. Now the interesting thing here is that these mammograms are high resolution, so you have lots of pixels that you have to produce, and at the same time the subtleties matter, right? Because in mammograms, it's very important to look at the subtleties, at the small patterns that you see in the images, because they will be important for later making a diagnosis, for instance. So why are we doing this? Now, obviously, I wouldn't recommend that we should use those images in any diagnostic task, right? But however, if you can fully understand how the imaging generative process works, you might be able to use those images for data augmentation. So you might be able to train your method by augmenting your training database. And this has been shown to actually lead to very, very successful results. So I told you in the beginning that deep learning has arrived, and I think that is clear. And I'm also very convinced that deep learning has come to stay. So what we see today is human-level performance algorithms. For sure, and they do already make an impact in clinical practice. You have seen the HeartFlow example, 20,000 patients being analyzed. Whenever we reach human-level performance with an automation, we can actually talk about being more efficient, being more objective. Right? That algorithm would make the same call 24/7 every day of the year, while radiologists might have a night shift, might have a different expertise, might have different levels of training. What I do believe is if we further move towards superhuman performance, that these deep learning technologies can really transform every part of clinical care, including diagnosis, treatment, therapy, and intervention. So with this, I thank you very much for your attention. Looking forward to any questions you have, and just want to add a little advertisement for a conference that we run next year in London. So if you're interested in deep learning for medical imaging, please join us in July next year in London. Thank you.
Thank you very much, Ben. We only have 5 minutes, so I'll quickly turn to questions. I'm sure there are loads because that was a fantastic presentation. Firstly, one conveniently at the front. Please introduce yourself first.
I'm Maithra. I'm a PhD candidate at Cornell, also research scientist at Google Brain. I'm also speaking next. So thanks for a really, really interesting talk. I had many questions. I'll stick to 2. So first question was, when you were talking about going beyond human performance, do you know that there's like a really big margin of error that humans sort of have? So like, are they sort of close-ish to optimal, or is it that in lots of cases they mess up, and are there even specific cases, like kinds of cases that they mess up in that we're trying to do better?
Yeah, so this heavily depends on the application. So the Royal College of Radiologists has released a report last year where they talk to errors in radiology tasks of up to 20 to 30%. It really depends on the task and what they are doing, but there is a huge variability. It might be sometimes they would vary within a few percent and they would always get it right in simpler tasks, but there are certainly radiology tasks which are very, very difficult. You have a high variability. So it's not that difficult to match that performance, right?
Right.
So you need to, of course, carefully check which areas that are, when you say, superhuman versus human-level performance.
And can you get to some of this superhuman performance by aggregating multiple humans? So you talked about having this panel that was sort of evaluating your model, but have you done things like maybe have multiple humans?
Absolutely. So two answers to this. You can aggregate on two sides. You can aggregate multiple human input, which hopefully will average out some of the variability, and there are specific techniques for doing that. You can also aggregate on the model side.
Yeah.
So you actually might train different models. They might capture a different aspect of the problem, and we have shown this. So the brain tumor segmentation challenge that we won last year, we actually used 12 different deep learning models. We aggregated them into like some sort of expert model, and that worked really well.
Okay, thank you.
Sure.
Cool. Right, we've got one there, I think.
Thanks for a great talk. This is Finn Catling. I spoke earlier today. I was just interested with respect to the heart model. Have you done any comparisons between your modeling and functional imaging, myocardial perfusion scans, that sort of thing?
Yeah, so actually in the trial, in the Pacific trial, they did exactly this. So they compared other non-invasive techniques, including functional imaging. So there was SPECT and others. They do a slightly different thing, so they don't directly try to estimate this fractional flow reserve, but all the other non-invasive techniques were worse than the heart flow analysis that tries to actually kind of mimic or simulate the gold standard of going into, with a catheter, doing the measurements.
I'm going to try and find someone who hasn't spoken yet. We've got maybe one there. And if you could be quick so that we can maybe get one more question in before going on to Mithra.
Chris Warwick, product development consultant from Sagentia. I was going to ask how much more development is needed before these kind of image segmentation techniques can be used kind of live for intraoperative CT, for example?
So again, it depends on your needs in terms of accuracy. And it depends on the imaging that you use intraoperatively. There are certain techniques that are real-time capable, so it's not a problem about computation speed. It's more the question, do you have the robustness, right? And I guess the other question here in the surgery, in the interventional setting, what you really want is to have a system that can tell you when it fails. So I think there is more research needed on the methodology side in terms of machine learning. How can we make sure that the models are robust, and how can we trust them in every case, right? And of course they will fail, but then we need to know. And during surgery, you want to know if your automated measurement is wrong. The doctor can go back to a fallback solution, but you have to have these techniques in place. And I think there is still quite some research needed.
I think if we're very quick, we can get through one more.
Thank you. I'm Saonni from Korea. I have two questions. One is that for the clinician, the size is also important, but the ratio of the size growing is much more important for the prognosis. And the other is when the position of the tumor is most important for the prognosis. So do you have any plan to study about that or have some,
Yeah, so in terms of how those measurements that we can provide with the technology, how they are used clinically, is a bit of a different question. I'm not a doctor, but what we are providing here are new ways or better ways of actually taking measurements, right? So currently, if you look at the brain MR with the tumor, doctors, of course, can know the location and everything. They do their planning for surgery or for radiotherapy. But here they get extra information. How they are incorporating this is clinical research, right? And how those techniques will affect patient outcome is a completely different story, right? So here we're only working on having a new tool to take measurements. What we do with those measurements is something we have to see exactly. Yeah.
great. I love that we had a kind of mini simulated panel discussion with all the speakers. And so what's great is that I can now thank Ben, and I'd love you to join me in that, and introduce to the stage, this is Maitra Raku. She is a research scientist at Google Brain, and she will be talking to us about direct uncertainty prediction for medical second opinions. Thank you very much.
Thanks. Oh, is this on? Okay, great. So hi, everyone. My name is Maitra. I'm a PhD candidate at Cornell University and also a research scientist at Google Brain. So today I'm very excited to talk to you about our work on direct uncertainty prediction for medical second opinions. This is joint work with multiple wonderful co-authors across many institutions, Cornell, Harvard, and Google Brain. So I'm going to start with a scenario that I think we can all relate to, which is, suppose somebody wakes up and they've been feeling unwell, and they're not sure exactly what to make of their symptoms either. Maybe they have shortness of breath, or blurred vision, or chest pain. And in all of these cases, they decide to go see the doctor to try and understand what's causing them to feel this way. So they get to the doctor, and the doctor's going to find out a little bit about their background, their medical history, and also listen to their symptoms. Based on all of these, the doctor might suggest additional tests for the patient to take. And finally, aggregating all of this information, they're going to suggest a diagnosis and a particular clinical course of action. So are things resolved? Well, unfortunately, not in all the cases. As we've been seeing in some of these talks, tests and symptoms are often difficult to interpret, and even for the experts. And there are many studies that have been done that show that different doctors, when looking at a certain patient case, might actually end up giving very different diagnoses. One recent study that came out just along these lines last year was a paper by Mayo Clinic, April last year, where they compared referral diagnoses to final diagnoses. And what they found is that only in 12% of the cases was there exact agreement between the referral diagnoses and final diagnoses. And more concerningly, in 21% of cases, there was a significant disagreement. What this often results in is that this group of patients with very different referral and final diagnoses also becomes the most expensive to treat. When we look at these statistics, the first natural question that comes up is, well, what's the source of all of these disagreements and uncertainty? Is it that, the doctor has just been a little tired or maybe slightly careless and there's just some random mistake that crops up? Of course, it's not so simple. Oftentimes, what we see is that certain patient cases have features that genuinely make them more confusing for the experts. So just taking an example from our data set, here are two patients in our data set, and in both cases, we asked the doctors to sort of go away and individually vote what condition they thought the patients had. And so for one patient, patient 2, we see that all the doctors agreed on the diagnosis for the patient. Whereas for patient 1, there's a huge range of opinions in what diagnosis the patient had. We can appreciate this further with an example with chest x-rays. Apologies to the doctors in the room for whom this might be very familiar. A chest x-ray is a relatively common additional test a doctor might suggest a patient takes for symptoms as varying as chest pain to shortness of breath. And so here are two chest x-rays from two different patients, and what we can see is that despite the fact that there are some differences, here the rib cage shows up more clearly, and this person has some metal implants. I don't think you can see that quite so clearly here. There's a lot of structural similarity between these two images. However, the ground truth conditions, the diagnoses for these patients, are very different. This patient has atelectasis, which is a condition where the cells in your lungs have been deflated. And this can be quite benign in many cases. It might just be a result of over-exercise or maybe even exhalation. And sometimes the only recommended clinical course of action is to do deep breathing exercises and sort of simple treatments like that. On the other hand, this patient actually has lung cancer. And this arrow here is pointing to a subtle feature in the image that actually corresponds to a tumor. And they, of course, need chemotherapy. Now, the question we're interested in is whether can we use machine learning to try and detect these subtle features in these images that might cause experts to be confused or disagree with each other.
In particular, the question we're interested in answering is whether we can train machine learning models to identify patient cases that are going to give rise to a lot of expert disagreement. These cases that the model predicts as giving rise to lots of disagreement can then automatically be flagged for a medical second opinion. This might mean that these patients who are at high risk of being misdiagnosed can automatically have their diagnosis corrected, or at least notice that this might require further attention. So the main medical setting that we're going to be looking at this in is with fundus images and diabetic retinopathy. So the fundus in the eye is the back of the eye behind the lens and includes parts such as the retina and the optic disc. And the fundus image looks kind of like this and can be used to diagnose you with different kinds of eye diseases. One of these eye diseases, despite being treatable, is also a leading cause of blindness, is diabetic retinopathy. Diabetic retinopathy, or DR, is graded on a 5-class scale. 1 corresponds to no DR, 2 is mild, and 3, 4, and 5 are moderate, severe, and proliferative DR. The grade 3 is also an important clinical threshold. 3 and above in DR is known as referable diabetic retinopathy, and 1 and 2 are non-referable. From a medical perspective, doctors want to make sure that patients with referable diabetic retinopathy are correctly diagnosed so that they can be treated and avoid going blind. So in this setting, we'll be looking at machine learning to try and predict cases of diabetic retinopathy that might result in a lot of expert disagreement. So we're going to have patients XI, and along with the patients, some associated fundus images. These fundus images are going to have multiple labels associated with them. And these multiple labels correspond to different doctors individually looking at this image and grading what it should have on the DR scale. On top of this, we're going to train a deep neural network, a convolutional neural network, to output a single scalar value, which is going to be a score between 0 and 1, and it's going to tell us whether the model thinks this image is likely to cause high disagreement amongst experts, or experts are mostly going to agree on the diagnosis. Now, for some of you looking at this diagram, you might realize that there's another way that we could get to this single score of expert agreement or disagreement. And that's, in this case, by first training a classifier. So diabetic retinopathy is graded on a 5-class scale, and so we might consider first training a classifier, which is going to output a probability distribution on these 5 classes, and using that output probability distribution to estimate the uncertainty we might see. So drawn schematically, it might look something like this. We have our problem instance or our patient, and from that we can either directly predict the amount of uncertainty or disagreement we might see, or we can first classify, and from that classifier try and predict the uncertainty we might see. Interestingly, what we find in practice is that for the application of determining when experts might disagree, the direct uncertainty predictors tend to work much better. There are a few intuitive reasons for why this might be the case. Firstly, it's actually very difficult to calibrate deep learning models. So there are sophisticated statistical techniques to try and ensure this probability distribution reflects the true probabilities of events occurring, but that's challenging. And additionally, what we suspect is that many patients have features that might tell us something about experts disagreeing, but not necessarily about the ground truth diagnosis. So before continuing, I want to mention one thing about our training data, because it has certain features that are common to lots of problems of machine learning in healthcare. So to train some of these large deep learning image models, you need quite a lot of data, even if you've pre-trained your model as we had done in this case. However, collecting a large data set in the healthcare space is very time-consuming and very expensive and very difficult. So our training data, while being relatively large, is also quite noisy. We only tend to have 1 to 3 labels per image, which is a challenge because sometimes, in fact, in 20% of our images, we see large disagreements amongst the labels that the image has. Nevertheless, we use this data set and we train our 2 classes of models, the direct uncertainty predictors and the classifiers, which we later use to output an uncertainty score. now that we have our two classes of models, we'd like to evaluate the question we're interested in, which is, can these models actually identify the cases where experts are most likely to disagree or get the ground truth condition incorrectly? But of course, our training set is pretty noisy. And as healthcare is a very sensitive and important application, we want to be dead sure that our model evaluation correctly reflects the actual capabilities of the model. And so to do that more accurately, we turn to what we call a gold standard evaluation set, an adjudicated data set. This data set is very small in comparison to the training set, and is also quite different structurally. Firstly, each fundus image now has many individual grades by specialists. So these are DR specialists who are looking at this image and have more insights into the kinds of features that might correspond to the different DR grades. Additionally, each image also has a single adjudicated grade. An adjudicated grade is something that's given to the image by a panel of experts coming together and discussing the important features they see in the image and coming to a consensus on the diagnosis that this image should receive. The point to note is that this data set actually has very different statistics to the training data we've been using for the models. And this is a challenge that machine learning models often have when there's a distribution shift between the trained distribution and the evaluation distribution. Nevertheless, the adjudicated set is very useful in enabling us to ask the clinical question of interest, which is, if we take the adjudicated grade to be a proxy for the unknown ground truth condition, so a patient comes in and they have some unknown condition, the question we're really interested in answering with our models is, can our models, which are maybe trained on noisy training data, correctly identify cases where an individual doctor, or your typical doctor diagnosis, is likely to disagree with the unknown ground truth condition? And mapping this onto our adjudicated data set, what we can do is we can see if our models can correctly identify the cases where the individual specialist, or maybe even an average of specialist grades, disagrees with this adjudicated grade we have. So the short answer is yes, they can actually do this pretty accurately, despite the distribution shift between the train and test data. And as I mentioned, we also found that these direct uncertainty models seem to perform better than first trying to classify and using that as an uncertainty estimate. In fact, we get pretty strong results supporting the latter. So the top 4 models here are models that first try and classify, and then we use the classification output as an uncertainty estimate, whereas the bottom 4 are different kinds of direct uncertainty predictors. The columns correspond to different ways in which we can evaluate these models. And what we find is that all the bottom 4 models do better than all the top 4 models in all of the evaluation methods. So that's pretty strong evidence that these direct uncertainty predictors are aptly designed for the TULSC. So that was our main evaluation that we looked at, but before wrapping up, I wanted to touch on a couple of other evaluations and applications of this methodology. So firstly, in practice, one thing hospitals find very useful is actually being able to rank cases. So sometimes doctors have a huge amount of cases or data to look at, and they'd like to know how they should allocate their effort. And so one thing we look at in our paper is, aside from the standard accuracy and area under the curve styled measures, we also looked at how well our models could rank the cases by cases of most uncertainty to least uncertainty. And there are measures such as Spearman's correlation that let you gauge this, and I invite you to look at the paper for more details. Another thing that's worth mentioning is that most of this talk has been looking at the medical applications of having a machine learning model that can predict when experts are likely to disagree. But in fact, this kind of a predictor has a lot of very useful applications within machine learning itself. So as I'm sure many people here can say, the most important part of training a good machine learning model is having a good data set with high quality labels. And the way these data sets are collected, even within machine learning, be they image data sets or language data sets or speech data sets, is they're done via crowdsourcing. So different data points go out to many individual human labelers who will look at this data point and provide a label. Often these are noisy and have to be aggregated. Additionally, this entire process is very expensive, and there's typically a budget on how much labeling effort you can use for these different data points. Oftentimes, this labeling effort can be allocated sort of uniformly at random, so each data point will get an equal amount of labeling effort allocated to it. But instead, we might consider using model uncertainty predictions to guide how we do labeling for machine learning data sets. And what we found is that any kind of model uncertainty prediction guided labeling effort could lead to very large improvements, say 5 to 10% improvement on ground truth agreement under the same budget. So in conclusion, in this talk, we've seen that machine learning has a lot of potential for identifying cases where experts might disagree with each other. And methodologically, direct uncertainty predictors seem to have a specific aptitude for doing this over a classification-based uncertainty prediction. And finally, while I mostly focused on healthcare applications of this kind of methodology, there's also many potential applications within machine learning itself, especially for data labeling, collection, and curation. With that, thank you, and happy to take questions.
My pleasure to announce Stephen Finbeiner. He is Director of the Center for Systems and Therapeutics in the Tolbe or Corret, excuse me for my pronunciation, Center for Neurodegenerative Disease at the Gladstone Institute, and he'll be talking to us about applications of deep learning to neurotherapeutic development.
Hey, thanks, Lydia. Yeah, so I wanted to talk to you today about some of the work that we're doing trying to use deep learning to gain insights into causes of neurodegenerative disease and finding treatments. And it's really a huge problem. This is a demographic curve. It happens to be the U.S. population, but it could represent Europe as well. And there's this big jump here which represents the baby boom. And this is a big problem for neurodegenerative disease because the number one risk factor for these diseases is aging. And that's Alzheimer's, Parkinson's, Huntington's, ALS, currently the number 5 killer. And it's a lot of people predict it's going to just be a tsunami that hits our healthcare system, and we're not really prepared for it. And it's not just a North America and European problem. People estimate that there are more people in China by 2040 with dementia than the rest of the developed world combined. And it's still the case that as of 2018, not a single disease-modifying therapy exists for any of these major neurodegenerative diseases. So a lot of people think it's sort of the healthcare challenge for our generation. The good news is I think that there are opportunities for deep learning at almost every step along the process of doing drug development. So people estimate it costs about $1 billion, maybe $1.5 billion to make a new drug. And there are steps along the way from discovery to preclinical development all the way to clinical trials where I think deep learning may have a role. Now, this year saw the 190th consecutive failure of an Alzheimer's disease clinical trial. And people think, maybe the brain, the most complex machine in the universe, is a really tough nut to crack. I think some of these tools, though, may have the power we need to really unravel some of these things. And I'm going to try to give you a few examples of how we're trying to do it at each of these stages. So getting more from the data that we collect, maybe giving us new ways to be able to uncover targets and find therapies, and then even maybe ways to construct clinical trials better. So the first example I'm going to give you is one that we published earlier this year in Cell. This is a collaboration with some engineers at Google. And what we were trying to do was just take microscopic images. So these are kind of grayscale images of cells. And to be honest, people in the biological field haven't really used these images for maybe a couple decades because they thought they could get basically all the information that was available just by looking at them. And so what people have gone on to do is to kill the cells, stain them with various dyes or other ways to label the cells to see different structures that normally aren't visible to the human eye. What we wondered was maybe there's more information in these images than people actually think. Maybe we can create training data sets and teach a neural network to be able to take images like these and to be able to predict labels that normally humans can't see. And so we created a UNet deep learning neural network, created a large set of training images. We took images from 3 different laboratories because we wanted to avoid overfitting our model and to be able to develop something that was generalizable. And then we looked to see what sort of predictions we could make. So this was done with Google Brain. I'm from San Francisco, and so the group is just south of us. This is what a typical grayscale image looks like. These little bumps here would be cell bodies, and these are neurons. And the first thing we wanted to do is to see if we could predict where the nucleus was. So that's something that normally you don't see except by using a special dye called DAPI or Hurst. And you can see it here. This is the actual label from this image. And after training a neural network, we were able to get it to be predictions that were about 96% correct. And we probably made this task more difficult than it needed to be by asking the neural network to make a pixel-by-pixel prediction of whether there was a label there and actually how bright it was. And so with this success, we tried a bunch of other labels. So things, labels that look at different cell structures, part of the cell's cytoskeleton, different labels of nuclear factors, other sort of cell type labels. And in each case, it was possible to train a neural network to take an image of live cells and to make predictions that a human wouldn't be able to make except by fixing these cells and labeling them with these special dyes. We took it a step further, so this is a picture of cells from the brain that have been dissociated and plated in a dish. And so there's a mixture of neurons and non-neuronal cells here. And typically for a human, it's really difficult to just look at an image like this, even a trained neuroscientist, and to tell which cells are neurons and which aren't. So humans usually have to use labels that can only be seen after you fix and stain the cells. But we were able, with enough training data, to be able to generate images that predictions from the neural network that looked virtually identical to what the labeled images looked like. And we've made these algorithms available on GitHub publicly. I think the take-home lesson from this, certainly for us, was there is a lot more information in these images that we collect than we thought. And by creating these large algorithms where we can train on training data sets that would normally require expensive reagents to be able to collect the labels, we can basically make predictions from images like these to be able to look at a lot of different biology without having to do this step. I think, so that's an example of how we can get more data, more information from the data that we collect just in the discovery phase of developing neurotherapeutics. How about finding causes of disease? So one of the challenges is developing good human models of disease. We rely on a lot of other systems, but a couple of years ago there was a breakthrough discovery where we were able to use skin cells or blood cells and to be able to introduce some genes to turn those into stem cells, and then with recipes convert those stem cells to various other types of cells in the body. So these are beating cardiomyocytes, so heart cells that were just fibroblasts a few weeks earlier from the patient that we collected them from, and we can do this with blood too. It's really useful for neurodegenerative disease because most patients don't want to give brain samples. So being able to collect blood or skin and to be able to make neurons out of them is terrific because now we can, for the first time, have a model of a human disease from a patient, for example, who has an Alzheimer's disease. So then the issue is creating ways to be able to generate lots of training data sets. So we've built systems called robotic microscopes where we have the cells grow in a robotic incubator. We've integrated robotic arms to transfer plates to and from the incubator, and then an automated microscope to be able to collect images around the clock so that we can, in a very standardized way, be able to collect training data. We also have programmed the instrument so that it knows where that plate is in three-dimensional space. And what that allows us to do is to go back to exactly the same set of cells as often and for as long as we want. So this happens to be a random image collected once per day for 7 days, where the plate goes back and forth in between. And hopefully you can appreciate how finely aligned the images are and how it's possible to follow a single cell, much like you would a patient, over time, and to be able to use these longitudinal data. So one of the things I said we were interested in was neurodegenerative disease. It turns out it's really hard for humans to accurately tell when a cell has begun to undergo neurodegeneration. We know this because we have created portals to display images to people in our own lab and have even displayed the same image on different days to see how accurate people are. The best they can do is roughly 80%. So one of the things we did to try to establish a better ground truth than human curation was to create a biosensor that's green when cells are healthy and it changes yellow when cells begin to irreversibly commit to a fate of neurodegeneration. And we wrote computer programs to be able to take the data off those robotic microscopes, carve them into portraits so that we could create large-scale training sets from experiments like the ones I showed you. So in this case, we used a training set of 23,000 examples. The cells on the left are ones where the ground truth's been activated, and the ones on the right are where the cells are healthy. And we used a convolutional neural network to try to train the CNN to be able to make a prediction of whether the cell was undergoing neurodegeneration or not just by the shape change. So this is something humans could only do about 80% of the time correctly, and that's after decades of trying to improve their predictive power. And so after setting up this CNN and making the prediction, within 3 hours we could get a CNN to make a prediction at 95%. And with just modest additions of a support vector machine, we could get it to 99. And we've used this now on rodent cells, human cells, organoids, in vivo cells, and it's a very robust prediction. So this has been very helpful for being able, from series of images like the ones I showed you, to define when neurodegeneration begins. And then another way we can use deep learning is being able to follow these cells over time, and then being able to use deep learning to predict when neurodegeneration begins, now to try to see if we can go back in time and find something about an image of a cell that's going to exhibit features of neurodegeneration, but find out the earliest event. Because what we really want to do is be able to intervene at the earliest possible stage. And so can we make a predictive connection between changes that a cell exhibits early in its lifetime that would predict whether and when a cell exhibits neurodegeneration. And I'll just summarize a lot of the effort. So we've been able to generate these dynamic images of cells undergoing neurodegeneration, models of Parkinson's, Alzheimer's, ALS, and other disorders. And what we've been able to find is that in many of these diseases, there are proteins associated with these disorders, like amyloid or synuclein, that are prone to misfold. They form toxic structures in cells that can cause deleterious effects. And in some cases, these form deposits that we think actually help mitigate the problem, at least temporarily. But what it really pointed to was that if there was a way to boost the ability of the cells to refold misfolded proteins or to clear them altogether, that could be therapeutically helpful. And so I think one of the utilities of this sort of deep learning approach to being able to follow cells longitudinally and to be able to find features that predict the future fate of the cell is we can come up with a blueprint for what might work as an intervention. And based on this, we started to develop small molecules to stimulate a protein clearance pathway called autophagy several years ago. And just this year, spun out a company called Caspian Therapeutics. And I'm a founder of this company. But these small molecules mitigate neurodegeneration phenotypes in a number of the human disease models that we study and are able to get into the brain. So we're hopeful that approaches like this will be able to help us identify important places where we might be able to intervene and then come up with strategies to do so. The last example I'd like to show you is just an idea about how deep learning and AI may help us to do clinical trials better. So right now, when we see a patient in the clinic and they have a memory disorder, we may give them a diagnosis of Alzheimer's disease, or maybe they have a movement disorder and we diagnose them as Parkinson's disease. About 90% of the time, those patients have no known genetic mutation. They have what's called sporadic or idiopathic disease, which is just to say we really don't know what's causing their disease in that case. And so we can't even be 100% sure that every patient with Alzheimer's disease or Parkinson's disease actually has the disease for the same reason. It's conceivable that even though we call patients Alzheimer's disease because they resemble each other clinically, it may be that at a molecular level, these patients, subsets of them, actually have diseases for different reasons. This has been shown to be true for cancers, and it's really opened up the door, I think, for being able to develop targeted therapies that have been very effective. For neurodegenerative disease, it's sort of a nightmare scenario because if we don't know which patients might benefit from a particular therapy, we're forced to test it on patients that are unstratified. And because these trials are so expensive and we have no way of knowing who would benefit beforehand, we may have actually a small molecule that works in a subset of patients, but we'll never be able to demonstrate it because the trials simply aren't powered to be able to detect it. So we badly need new ways to be able to tell which patients might represent a subset that would benefit from a different therapeutic strategy. So one of the things we're trying to do now is use deep learning to diagnose disease in a dish, basically. To take cells from patients with different diseases, especially sporadic diseases, and then to see if we can train a deep learning network to be able to predict which disease that patient has, and maybe even to see whether that the features of those cells cluster in any way. We've begun and have had some success using cells from patients with spinal muscular atrophy compared to healthy controls, where we've been able to get diagnostic accuracy around 90%. And these are in cells that really, to a human eye, we've not been able to see any difference, but we can train a neural network to be able to make distinctions. And we're now in the midst of a very large clinical study where we're enrolling 1,000 ALS patients, doing very deep clinical phenotyping around them, generating those stem cells from every single patient, and then we're going to use those to make motor neurons to be able to basically do the same thing. To find out, is there anything that we can measure in a dish that predicts what happens to that patient in the clinic? Because ultimately, being able to predict whether or not something's going to work for patients in the clinic would make a huge difference in terms of really de-risking the whole area of developing neurotherapeutics. So hopefully you get a sense of just a sample of some of the ways I think deep learning could really make a difference in developing neurotherapeutics, from being able to get much more information from the data we collect in the lab, to being able to do things like predict the future and be able to unravel complex cause and effect relationships in cells that could point us to targets. And then ultimately being able to, in an unsupervised way, look at relationships between things we measure in the lab and things we measure in patients that might point to productive ways to be able to study them. So I'll just end there and just highlight a few people who've made really big contributions to this work. Jeremy Lindsley was really a leader in our group for developing some of the ground truth biosensors around cell death and being able to train and develop computer vision convolutional networks to be able to do those predictions. A great group of Google engineers who helped us with some of the initial work on being able to predict labels in cells from unlabeled images. So thanks very much for your attention.
Thank you so much, Stephen. That was fascinating. And I expect that there are a lot of questions. I would hope so. Okay. If not, there we go. One over there. Please introduce yourself first.
I'm Sarali from UCL. I was wondering if you could expand on the use of inducible stem cells in your approach. Is it to just get an understanding of how cells develop in that person, or are you using it for a therapeutic purpose?
Yeah, so these are induced pluripotent stem cells. So they're stem cells that we make from adult cells from those patients. So it could be blood cells or it could be a skin cell. And we can introduce genes into those cells to turn them into stem cells, then with various factors, turn them into virtually any other cell in the body. So generally speaking, the strategy is trying to make a model of the disease that they have by turning that stem cell into a cell that's susceptible in the disease that they have. So for Parkinson's disease, it might be a dopaminergic neuron. For ALS or motor neuron disease, it could be a motor neuron. And so once we have those in a dish, we can form either really simple models where we just make the cell itself, or more complicated organoids where we're building, multiple cell types together that may actually have some anatomy similar to sort of a tissue that a patient may have. But then with imaging, you can follow those over time, so you can begin to see features of disease appear once those cells are differentiated into their mature cell type. So we and others have been able to demonstrate, for example, increased neurodegeneration, other features that you might see in the human disease. So being able to use that as a way to model disease that the patients have as a way to be able to both explore what's causing those features to appear and to figure out if there are ways you can intervene to prevent that fate from happening.
Regenerate cells in the environment, but not in the central nervous system. So how do you do that when you organize it?
Yeah, so it depends on the model that you're trying to create. So there's frequently a trade-off. The more complex the model is, the more variability you get, the more difficult it is to generate data that is useful for screening, for example. So I think one really important question is, how complex do you need to make the model to make it useful? And it's not always clear-cut, I think, what the answer to that question is. Sometimes just a single cell type is enough. Sometimes you need to build more complex structures to be able to see certain biology or even get cells to mature enough. But it depends a little bit. So some cell types require such different recipes to make them that you sort of have to make them separately and then put them together. In other cases, for example, we've made a neuromuscular junction, so we've got a recipe that makes cells into a motor neuron and a skeletal muscle that form a connection and contract. And so sometimes you can, with just a single recipe, make multiple cell types that will form interactions that are similar to what you might see in people.
Wow. Okay, we've got another question at the back.
Thanks. Matthew Fennick from Future Advocacy. That was a fascinating presentation. I wonder if you could comment about getting these tools sort of incorporated into the pathways. As you know, doctors, clinical researchers, scientists are and paper reviewers predominantly are a pretty conservative bunch. So how do you get these tools sort of integrated? How do you get them accepted? Has it been something where people have jumped at the opportunity to use them, or is it about convincing the community?
Yeah, it's a great question. I think my experience so far has been that there's a really big divide between biology and computer science, and it's very difficult to get biologists who aren't that familiar with these tools to feel comfortable to adopt them. I think in general, they're pretty surprised at what you can do with them, and they dearly would love to implement them, but they don't themselves have the expertise to be able to do it. So I think my suspicion is just seeing kind of even my own group, where I have biologists who, once they see firsthand the power of some of these tools, have begun to latch on to the computer scientists and begun to learn to code and do things themselves. And so I sort of see just I think it may be a generational thing where people are going to have to see it for themselves and begin to acquire it. And I think maybe as time passes, you're going to see scientists who have skills on both sides. In the meantime, I think we try to make the code that we generate available publicly. So we have, for example, a Fuji plugin. We made a deep learning network to be able to tell whether images were in focus or not without having anyone change the threshold. So that's a pretty simple Fuji plugin. It took a little effort to set it up, but that should be really easy to implement. And we are aware of people trying to implement some of the code I showed you earlier on the GitHub. And, as you might imagine, we've had to help in some cases give people support and stuff like that. But my hope is that, slowly people will adopt these. But it's a great question. It's a big challenge. Yeah.
Great. Any further questions? Everyone's very keen for lunch, are they? Okay. I think then, thank you very much. Details for lunch. So after this session, you'll be merging with the Deep Learning Summit in the other room. So make sure